So everyone ready? I just want to start. Okay, wonderful. So good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, and annyeonghaseyo. And hello all. Welcome and thank you for joining us the 31st seminar, and I call MIT Day 3 because we have now previously two MIT professor, and then we have another joining us today. I'm so excited to meet many SynBYSS people next week in person during the SEED conference in Washington, D.C. and SIMB conference in New Orleans. And this will be my 7th in-person seminar or conference this year, but I want more. So another thing I want to say today is thanks for your joining, or the audience and speakers, especially during the busiest time of the year with the final exams and semester ending right now. Actually, my colleagues said, they cannot join us today because of their, another commitment, especially the, exam kind of giving and so on. So, okay, it's my tremendous honor to introduce very briefly our pioneer speaker, Professor at Boyden. He does not need any introduction because we all know him. He is the Y.Eva Tan Professor in Neurotechnology at MIT and an HHMI investigator. He's also a National Academy of Science member, AAAS member, an NIH Director's Pioneer Awardee, a Breakthrough Prize in Life Sciences winner, to name just a few. And without further ado, Professor Boyden, the virtual podium is all yours now. And thank you so much for your time and excitement. Thank you.
A little 5-minute mini lecture. And I thought a lot about what I could squeeze into 5 minutes that would be of interest to this wonderful broad community. And of course, as synthetic biologists, what we're trying to do is to invent technology that can help us understand or repair or build from scratch things of great importance to us. And of course, the way that many classical engineering disciplines are defined, like electrical engineering or mechanical engineering, is that you learn skills that you then practice as your trade. And so I started wondering, what is happening in bioengineering? Are we converging? Are there strategies that we can use day in and day out to solve problems? And I think if you look at what we struggle with in bioengineering, the answer is that it's a lot harder, right? If you're smart and you work hard, you can design an electrical circuit in a computer, you can test it in a simulation, you build the circuit, and quite often it works. But in biology, there's so many unknowns, right? There's so many building blocks of life, right? The human genome contains tens of thousands of genes, encoding for who knows how many biomolecules. There's so many interactions that we don't even know how to begin sometimes to understand the impact of a tool. And so in fields like electrical engineering or computer science, you sometimes see that you can be very smart and work hard and you get it done. But in bioengineering, it often seems that you can be smart and work hard, and then a bad stroke of luck hits in year 25, and now what, right? So I started thinking about our group. Our group tries to build inventions like optogenetics, where we can control brain activity with light. We have a method that we call expansion microscopy, where we can take physical objects like cells or tissues, and we can chemically grow them to make them 100 or 1,000 or 10,000 times bigger in volume. It lets you do nanoimaging on ordinary microscopes. And I started wondering, are there principles that might emerge that allow us as bioengineers to continuously innovate, to generate new inventions, and avoid, ideally, a stroke of bad luck, killing things off late in the game? And I thought back to a moment in my life, which was very gratifying, which is what got tenure at MIT in 2014. And my research group commissioned Post-it notes of things that I would say too much. So I could just put the Post-it note on a piece of paper rather than having to talk about it. And I thought I'd pick 3 of them today and show them to you because some of these I think have stood the test of time and are interesting to think about. So one of these that my group commissioned, a Post-it note of something I say too much, is to work backwards from the goal. We want to build technologies that solve problems. And that means we have to think about the problem and understand it. I think in technology fields, it's very common sometimes in some fields for people to try to build a tool, but not necessarily know what the goal is. That doesn't mean we should shy away from serendipity. Serendipity, of course, has yielded huge innovations, ranging from CRISPR to fluorescent proteins, the list goes on and on. But if we have at least an awareness of the problems in mind that could be solved, that can guide us on what we can do. You know, with optogenetics, we were driven by this goal of repairing aberrant brain activity. For expansion microscopy, you know, the building blocks of life are very small, right? Biomolecules are very small. Why can't we help everybody do nanoimaging? And so having a goal in mind, at least in the back of your head, can help guide our technology inventions so that they can have the maximum impact possible. The second thing that my group commissioned on these little Post-it notes is it ground truth? Now, what does that mean? Well, the history of science sometimes shows these moments where enormous productivity happens. Chemistry after the periodic table, physics after quantum mechanics. So chemistry and physics existed before the periodic table and before quantum mechanics. But there were moments in chemistry history and physics history where suddenly the application and technological progress became exponentially accelerating. After the periodic table, you could design polymers, you could build Teflon, you could build nylon, you could look at the structure of DNA, the list goes on and on. Before chemistry hit ground truth, the building blocks, atoms, and their connections, bonds, chemistry still existed, but it was slow-paced, right? Physics, after quantum mechanics, which again is sort of ground truth, right? The building blocks of atoms and how they interact through their forces, right? And now you have computers and cell phones and lasers and the internet and so forth. So I guess the interesting question that guides a lot of our thinking is, what's ground truth for biology? It has been hit a few times, right? Like the genetic code. But what's ground truth for the brain? What's ground truth for a cell? And so when we build tool sets like optogenetics or expansion microscopy, we are trying our best to get down to the fundamental building blocks of the brain and of cells and how they interact. Have we actually reached them? Well, only time will tell. But I think this idea of looking for ground truth is a very powerful way to make sure that we're pushing to the most fundamental descriptions of our field. And the third one, never make assumptions. Now, it's dangerous to say never, but we can try not to make assumptions. So many times in biology history, when people made an assumption, they later found out there's an exception. That's almost how evolution works, right? Evolution's trying to, exploit every possible way of doing something, right? Even the central dogma of biology, right? DNA yields RNA yields protein. But now we know there are retroviruses, which convert RNA back to DNA, and so on and so forth. So one of the things that we often try to do is to ask ourselves, what assumptions are we making that we might want to fix? And so one thing, of course, that is just so overwhelming is just the complexity of all the building blocks of biology and how they interact. If we don't know that two things interact, and if we don't know that they don't interact, how do we know for sure that later that interaction won't mess up what we're trying to build? And this is more of a meta principle than an actual action. But as we build tools, we're often wondering, is there some hidden assumption we're making that we should anticipate? And if it's something we can anticipate, that might help us make a better tool. If we can't anticipate it, maybe we should build a tool to find that out, because as the history of biology has shown, very often when we make an assumption, we later regret it, because biology is very complicated and lots of things happen throughout biology. And assuming that the problem is simple does not make it simple. And so, yeah, I'll leave you with those messages. And our group builds lots of technologies, and we're always eager to hear from people to learn about what problems they have, right? We always want to learn about other goals people have so we can help solve problems. And we love to build tools with other people as well. We co-develop technologies with people all over the world. Because again, if we're going for ground truth and if we're trying to avoid assumptions, it's not just interdisciplinary research, it's omnidisciplinary. Every field of science could have impact on revolutionizing biology and medicine for the better. And so why don't we make it happen? Stay in touch.
Wow. Oh my God, I was amazed by your talk. This is one of the best 5-minute talk I ever heard. I mean, I absolutely amazing. Thank you so much. And also, because of so many challenges we have, we are more excited because challenges are our opportunities. And I also absolutely agree with you. And I'm always asking a question, what is the governing principle in biology? And as physics kind of has, and I want to know the answer in my lifetime. So hopefully, thank you so much. Absolutely brilliant talk. I mean, amazing. Thank you. Okay. Thank you. Oh my God. So now the main speaker of today with a longer introduction, Professor Xiao is an assistant professor of chemistry and bioscience at Rice University. He obtained his undergrad degree from the University of Science and Technology of China, where he graduated with a bachelor degree in chemistry and honors degree in physics science. Han joined the PhD program at the Scripps Research Institute. His thesis work with the Professor Peter Schultz focused on expanding the technique of genetically incorporating non-natural amino acid in both prokaryotic and eukaryotic organisms and applying this technique for better cancer therapeutics. In 2015, he joined the laboratory of Professor Caroline Bertozzi as a Good Ventures postdoctoral fellow of the Life Science Research Foundation at Stanford University. In his postdoctoral work, he was engaged in the development of novel cancer immune therapy targeting the cell surface glycans axis of the immune modulation. In 2017, he started his independent research at Rice University. The focus of his research is the development of various chemical biological tools allowing us to understand complex biological systems as well as develop novel therapeutic strategies. His awards include Level 2 Breast Cancer Research Program, Breakthrough Award from DoD, Maximizing Investigators Research Award for Early Stage Investigator from NIH, one of the prestigious kind of award from NIH, Norman Heckerman Welch Young Investigator Award, and CPRIT Faculty Recruitment Award, to name several. With that, of course, I mean, he's the true rising star, and then he's right now touring for his tenure kind of evaluation. So if audience, YouTube audience watching this video, and then considering writing letter, I believe you should write one of the strongest letter for his tenure case. So with that, Han, take it away. It's absolutely my great honor to have you today. Thank you.
Thank you, Tae-Sok, for the wonderful introduction. And thank you so much for what you are doing for our young investigator. I think it's super helpful. And also, I feel, as we mentioned before, this symposium is already beyond a symposium itself. It serves a really important education role. I think today, Dr. Dalton's speak, this 5 minutes talk is a great example about inspiring new generation and what we should do, what kind of question we should target, and how we can do great science. I think that's all very helpful for the young investigator. So to start my talk today, the topic is generation of unnatural organism with a 21st amino acid. Before I start the topic, I want to briefly introduce what we are doing at Rice University. In my lab, we have 3 directions. The first part, we develop different imaging reagent that allow us to perform some super resolution imaging or some animal imaging. And for the second part of my lab, we focus on the translational medicine or different therapeutic. Recently, we work on this antibody molecule. So antibody and antibody derivatives, including this antibody drug conjugate or bispecific antibody and a checkpoint inhibitor, they have been serve a really powerful platform for the treatment of the cancer, infection disease, inflammation, or different immunological disease. And recently, people have used this antibody for the treatment of the COVID-19. So in 2017, when I joined Rice University, I started to think about, is there any limitation or this technology has been quite successful? Is there any other things we can contribute as a chemical biologist? So the problem we try to address for this drug is kind of biodistribution of this antibody. So antibody people claim it's a magic bullet, should only target cancer cell, but in the real world, it's not true. If you look at this biodistribution for the gastric cancer, this is an N87 xenograft model, and this cancer cell express the antigen called HER2. So after that, people will inject different isotope labeling anti-HER2 antibody, and they want to look at the biodistribution of this antibody. And what you can see is antibody is definitely target the tumor side and enrich over there. But at the same time, we will see the antibody is also go to our normal tissues, including lung, liver, spleen, kidney, and also the other tissues. So people tell antibody is magic bullet, but it's not 100% true. And you will see this kind of non-specific delivery of the antibody to the normal tissues. And this kind of non-specific delivery, they will cause this on or off target toxicity, which is the side effect we observe in the clinic. For example, this FDA approval anti-HER2 drug trastuzumab has been known for the fatal lung toxicity. And if we cannot deliver enough dose to the tumor side, it also causes the problem. People already show the not the optimal dose of the delivery of the antibody to the tumor, they can help the tumor cell to evade the antibody immediate killing. In that case, they can generate some resistance, they can generate tumor relapse. So from here, you can see it's very important if we can enhance the antibody biodistribution in the tumor side and reduce their biodistribution in the normal tissues. So how we can do this? So in my lab, we have a really easy hypothesis of the idea. We want to introduce a different specificity to the antibody. Right now, antibody has a tumor or antigen specificity. We want to introduce a tissue specificity to the antibody. In that case, antibody can specifically enrich the disease tissue, and then antibody can use their antigen specificity to find the cells or cancer cells and kill them. So we started this story from our bone tissue. One of the reasons is our bone is a very unique tissue, but the major reason is the bone cancer is a really bad disease. So this is an X-ray image of the breast cancer patient with a bone metastasis. And what you will see is all these hollows in her bone, and that indicates the cancer cell metastasize to this bone tissue. They stimulate the osteoclast cell to eat up the bone. And for this patient, they will suffer severe pain. And the really bad thing is their bone density get a dramatic drop. They will suffer from fracture, very common in the clinic. And this kind of bone metastasis is not really rare. For example, bone metastasis is the most frequent metastasis for the breast cancer patient and also for the prostate cancer patient. And it's definitely a lot of pain. They cannot move. They suffer from fracture. And if you look at 5-year survival rate, it's only 13%. At the same time, there's a lot of clinical trials suggest right now the antibody drug is not really working well for this patient with a breast cancer bone metastasis. So at RISE, we try to think about how we can enhance the antibody therapeutic efficacy against this cancer cell in the bone cancer niche. So as I mentioned before, we want to drive this antibody and enrich them in the bone metastatic niche. And we can do that by making this kind of bone targeting antibody, and we can conjugate these different bone homing moiety, for example, small molecule, bisphosphonate. They can specific chelate with our hydroxyapatite structure, the inorganic structure in our bone. Or we can use a really simple antibody engineering approach, just introduce negative charge residue to the antibody, and then this negative charge, because of the electrostatic interaction, they can home this antibody to the bone metastatic niche. The therapeutic efficacy is really good. If you look at the curve, this is a breast cancer bone metastasis, and then we are looking at their luciferase signal increase. And this curve is the wild type antibody. You see the cancer cell will keep growing, but for our bone targeting antibody, we can have a really good inhibition of the breast cancer bone metastasis cancer cells. At the same time, we can boom up the survival rate for these animals from around 30% to more than 75%. To look at the bone metastasis in the clinic, people can use the X-ray, but it's not a really high resolution. We can also use the micro CT to look at this bone lesion. This is trabecular bone and cortical bone. We isolate from the wild type antibody treated group. And you will see a lot of bone lesion on this bone, and the bone density get very bad. But for our bone targeting antibody treated group, the bone surface is smooth and the cortical bone density is good. So right now we try to move forward and push some of these drug hopefully into the clinical. Hopefully someday we can help these patients suffer with the bone cancer. And after I jump into this field, I really know these patients, they really don't have a good quality of the life, and they don't have really good medicine for this cancer cell in the bone. For the third part of my lab, we are working on the cell engineering. We try to add the 21st amino acid to these cells. So as most of you know, our cells, they use the 20 canonical amino acid to build up their proteins, different enzymes, and carry out this biologic problem. So there has been a long time question why we have 20 amino acid, can we have the 21st non-canonical amino acid? If with a new building block in hand, can we evolve? What will happen to the protein evolution? Can we evolve something or species or protein with enhanced activity, or we can evolve some medicine with the enhanced efficacy? So to add this 21st non-canonical amino acid to the cells, there are some requirements. We need to create a unique codon to encode this non-canonical amino acid. At the same time, we need to evolve different translational machinery to specifically recognize this non-canonical amino acid, but not their canonical building block. Including my PhD advisor, Peter Schultz, and a lot of the other people, they have done elegant, tremendous work on this field. They have evolved different stop codon, amber codon, or frame codon, 4-base codon, or even unnatural base pairs to encode this non-canonical amino acid. And right now, more than 300 different non-canonical amino acid has been incorporated by different bioorthogonal tRNA and amino acyl tRNA synthase pair. But if you look at this diagram, what is really missing from these cells with the 21st non-canonical amino acid? That is the biosynthesis of the 21st amino acid. So right now, when people carry out this genetic expansion or try to incorporate the non-canonical amino acid into the protein, they need to rely on the chemical synthesis of this building block and feed that to the cells. And from here, you can see there may be a lot of limitation from this chemical or feeding of the chemical synthesis, the building block. I know we have some discussion previously on this symposium about this limitation about feeding these different chemicals. But from my perspective, I think there are several, and one of the major one is the poor membrane penetration. So some people claim the suppression efficiency is low or genetic incorporation of this non-canonical amino acid building block is low. One of the reasons is a lot of these building block, they have charge, they are polar, they have a hard time to penetrate the membrane. And the other limitation you can think about is the commercial not available, and it's very hard to make from a chemical perspective. And the last one, I think, is a big one. It's about the bioavailability if you want to incorporate this non-canonical amino acid in a living organism. And people already show this non-canonical amino acid, they have a poor pharmacokinetics in the living organism. In that case, if you want to do this for the animal, for the multicellular system, you need to keep inject this non-canonical amino acid. It's not a really friendly or easy or sustainable way to doing this. So to avoid this kind of limitation, we can generate these cells with 21st non-canonical amino acid by introduce different biosynthetic pathway. For example, we can introduce different enzymes can make this non-canonical amino acid, followed by introduce the bioorthogonal tRNA and the amino isotRNA synthase can recognize this building block, and we can use the amber stop codon to site-specific incorporate this new building block into the protein. After this, you can see the cell, they will not rely on any exogenous feeding of the chemicals, and they will be able to grow by themselves and keep making these new proteins or new therapeutic proteins or whatever with the new building block and the modification. At the same time, when we start this idea, or people start this in the field, I will give you some of the example. It's very hard to find this metabolic engineering or metabolic or biosynthetic enzyme can make this new building block. There is several reasons. The major reason is a lot of these amino acid modified enzyme that are post-translational modified enzyme, or they are the non-ribosomal peptide synthesis. They don't recognize the standalone amino acid. They will recognize the amino acid on the protein, on the peptide. So in that case, we don't have a lot of choice to make this standalone non-canonical amino acid for the genetic code expansion. And another reason for the biosynthesis of non-canonical amino acid is hard is for the genetic code expansion, we need to achieve a relatively high intracellular amino acid concentration. And in that case, the whole machinery needs to be very robust. So because of this limitation right now, we only have 2 examples for these cells with the 21st non-canonical amino acid. The first example is a bacterial cell created by my PhD advisor, Peter Schultz. He started from crossmate and he introduced different Streptomyces biosynthetic pathway, and they can make this aminophenylalanine, and they can site-specific incorporate that into the protein. And recently we showed this aminophenylalanine containing protein that are also very helpful to achieve this site-specific conjugation because it's a bioorthogonal handle. In this year, I think the Kunjapa group, they also reported a paper, they can redirect this aminophenylalanine to biosynthesize this natural phenylalanine and incorporate that into the protein in a site-specific manner. The second example is come from Jason Chen. Jason has shown they can use the biosynthetic way to introduce this kinase and biosynthesize this phosphothreonine, and site-specific incorporate that to the protein and to do a lot of interesting work. But what is very interesting is if you look at their efficiency compared from the biosynthetic to the chemical feeding. For the chemical feeding, they can almost cannot see any phosphothreonine intracellularly. After feeding like 1 millimole from outside, they cannot see anything inside of the cells. But if you look at their biosynthetic pathway, they can achieve almost like 2 millimole level of the phosphothreonine intracellularly. I think this work is really inspiring this work is also show, okay, this is the way we can enhance the suppression efficiency of the genetic code expansion. And also this is the way we can create a much efficient cells, right, with this new building block. So inspiring by all these works, in 2017, we start to work on this field. We start to create the cell with the additional new building block. And right now, people only have the example for the prokaryotic cell. We also try to create the cells with this new building block for the eukaryotic cell. One of the first example we work on is the 5-hydroxytryptophan. We try to create a cell can make this 5-hydroxytryptophan. And because this 5-hydroxytryptophan, people show it can use as a bioorthogonal handle and also for the other application. To make this 5-hydroxytryptophan inside of the cell or intracellularly, the most easy way is we can start from tryptophan. And people show there is a different hydroxylase and they can carry out this transformation. And in the prokaryotic cell, they are using this MH4 cofactor. And to start this, we just want to do a screening for these different hydroxylase. And we create a suppression or GIP reporter system for this kind of screening. So we introduce a GIP gene with the amber codon TAG in the encoding frame. And after that, we also introduce the orthogonal 5-hydroxytryptophan synthase and also the tRNA. If there is a 5-hydroxytryptophan existing or available for these cells, and the 5-hydroxytryptophan can be loaded to the tRNA, and the tRNA can suppress the stop codon in the GIP. And we will be able to observe the full length of the GIP. You will see the cell will get glowing and fluorescence. At the same time, if there is no 5-hydroxytryptophan available or biosynthetic by our machinery, and what we will see is a truncated GIP. The cell will not fluoresce. So this can serve as a really easy way for the screening assay. So we screen different hydroxylase. People reported can carry out this activity for this tryptophan or different phenylalanine tyrosine analog. And we found this hydroxylase from Schizomonas. They can generate a 5-hydroxytryptophan containing GIP. And furthermore, we show this cofactor biosynthetic pathway is important. If we can make more MH4 cofactor for this bacterial cell, we can boom up the 5-hydroxytryptophan containing protein production level at very high. And finally, we can achieve a similar level as feeding of 1 millimole 5-hydroxytryptophan to the cells. And we can use the mass spec and SDS page to confirm. We can set specific incorporate this biosynthetic 5-hydroxytryptophan, for example, into the GIP in this case. And because 5-hydroxytryptophan is an electron-enriched strain, so they can serve as a bioorthogonal handle for the bioconjugation. So we can use these cells with a 5-hydroxytryptophan to produce the anti-HER2 antibody with this new building block, and we can perform the azo coupling to incorporate the fluorescence molecule to the antibody. In that case, we can generate this antibody fluorescence conjugate. And we can use this conjugate to specifically deliver or detect the HER2 positive cell, but not for the HER2 negative cells. So after finish this, we also think about, is there any unique feature or application we can use for these cells with the synthetic 5-hydroxytryptophan? And we are thinking about maybe we can create some living sensor cells. So for example, people show this MH4 cofactor biosynthetic pathway is very sensitive to the oxidative stress. And in the presence of the enhanced oxidative stress, the cofactor biosynthetic pathway, they will be reduced and the 5-hydroxytryptophan level will be reduced. And we can couple this 5-hydroxytryptophan production with the GIP protein production. In that case, we hope we can look at the oxidative stress of the cells in living time. And the result is showing we can do that. On the left, this is control cells or traditional cell. People feed the 5-hydroxytryptophan. And you will see in the presence of the oxidative stress or in the absence of the oxidative stress, we cannot observe any GIP production difference. But for our cells, they can make the 5-hydroxytryptophan in the presence of the hydrogen peroxide. We will see a GIP response, and we can quantify this response with the hydrogen peroxide level. And hopefully someday we can, using these cells to report this oxidative stress for the microenvironment of multicellular organism. I think that will be super cool. So to summarize this first part, we generate these cells, can make the 5-hydroxytryptophan, and the protein with the 5-hydroxytryptophan with these cells, we can achieve the bioorthogonal ligation. At the same time, I think we show a very preliminary application. We can use these cells or unnatural cells for detection of the oxidative stress in living time. Besides working on the 5-hydroxytryptophan, we're also interested to create some cells with the site-specific post-translational modification. So quite recently, we created this new species. They can have this sulfotyrosine as a new building block, and we can incorporate that into different therapeutic molecule. At the same time, we demonstrate we can expand these cells or this idea from prokaryotic cells and to eukaryotic cells as well. So tyrosine sulfation is a very important post-translational modification for different secreted protein and also for a lot of membrane protein. And people show this sulfation is important for the cell-cell interaction and also for the virus interaction. And at the same time, sulfation is an important PTM for the different inhibitor, for example, for the blood clotting, and also for the different cytokine, different signal pathway activation. And to study the biologic function of this sulfation in a site-specific manner, people have used the genetic code expansion, people create a different tRNA and a different amino acid tRNA synthase, and a lot of researchers, my colleagues, they have working on this field. They can incorporate this sulfotyrosine in E.coli, in eukaryotic cell as well. But when we look at their paper, most of the paper, they need to feed like 10 to 20 millimole sulfotyrosine to the cells. And when we look at this, we see this is our chance. This indicate maybe sulfotyrosine, they have a hard time to penetrate the cells. They don't have really efficient for the permeabilization. So in that case, we want to create such a cell with the site-specific sulfation, and also the sulfation is a very important medical modification as well. So to start, we want to find out a biosynthetic pathway for this sulfation or sulfotyrosine. People show there's different sulfotransferase. They can use a PAPS as a cofactor. And our first part of the work is figure out which enzyme can efficient offer or carry out this sulfotransferase reaction. So we decided we are using the same suppression GIP reporter assay with sulfotyrosine. The GIP will produce. Without sulfotyrosine, GIP will be truncated. So we did a screening for different sulfotransferase. People reported can be used for this phenolic subject. And unfortunately, we didn't see any activity for people reported or literature reported sulfotransferase. And this is a really kind of hard time for us because we didn't see any basal activity. If we see any basal activity, it's a little bit, we can do the protein evolution, we can do metabolic engineering, we can make them better. But if we didn't see anything, and this is really hard. And at that time, I think the COVID is over here, we get a quarantine, and my student and I, we try to have some brainstorm, and we also try to learn some new technology. And I read different literature. I figure out people have the different bioinformatics skills. They can help us to find out some new enzyme with novel function. And one of the important tools people use is the sequence similarity network. And I start to ask my student, can we build up this SSN or sequence similarity network for the sulfotransferase. And he did that, for this coumaric acid, and because we feel this substrate is very similar to the tyrosine. And this is the SSN. And within this SSN, we also found another enzyme which can use a tyramine as a substrate. And my student come up with a really bold assumption. They said maybe some of the sequence which is close to both of these enzymes, they may be able to recognize the tyrosine, because you have the carboxylic acid from one enzyme, you have the amine, the primary amine from another enzyme. So if we can select some of the enzyme between these two yellow spots, maybe they can carry out the tyrosine sulfation. And we are very lucky, we pick up around 30 different enzymes from this SSN, and there is one enzyme from IBIS, and they can generate a little bit of this sulfotyrosine containing GIP in our reporter acid. And after that, we just use different engineering or metabolic engineering strategy to make these enzymes better for this sulfation reaction for the tyrosine analog. So my student first screening different knockout strand. These knockout, they are important for the tyrosine generation or for the PAPS generation. And we found the knockout with this cis-H enzyme. This is the enzyme to degrade the PAPS. We can enhance the sulfotyrosine production. At the same time, we can introduce the biosynthetic pathway for the PAPS to the cells, and we can enhance their production. And we also found the sulfotyrosine transferase, the expression level is very important. You cannot achieve or induce this gene at a very high level or very low level. We need to control the expression level to achieve the optimum sulfotyrosine production. So just to give you some idea, this is a final system we get. And for the bar in the gray, this is the chemical feeding. You can see people we feed the 27 millimole to the cells, but for intracellular sulfotyrosine level is only 300 micromole. Compared to our biosynthetic sulfotyrosine, we can achieve almost 900 micromole. So it's very efficient from biosynthetic pathway, and we will see a similar trend for the sulfotyrosine protein production. Our biosynthetic pathway, they can easily beat like 10 millimole or 20 millimole chemical feeding. So all this work so far was done in E. coli. And to move this story further, we want to try some mammalian cells, see whether we can create the eukaryotic species with the new building block. We can introduce this sulfotransferase, the salt T1C1 into the Hike cell, and also we introduce the amino acid tRNA synthase pair to the Hike cell, and we use the GIP with the amber codon as a reporter. And for the chemical feeding, you will see without feed any sulfotyrosine, the GIP is truncated, so you didn't see any fluorescence. But once we enhance the level of the chemical feeding of the sulfotyrosine, we see the enhanced level of the GIP. For our biosynthetics sulfotyrosine cells, they can produce a really high sulfotyrosine containing GIP over there. After finishing this work, and we start to ask a kind of biochemistry question. So why this sulfotransferase from IBIS is so unique? What kind of biochemical or molecular biology perspective we can provide it? So that's why we collaborate with my really awesome colleague, Peter Warnings, who is a great therapeutic chemist. And also his student, Shikai, is a really smart graduate student. They used the AlphaFold tool to predict the structure for this sulfotransferase from IBIS. And after that, we can dock the tyrosine to this structure. And you will see very easily this glutamic 1,6,8,1, they have a really important interaction with the tyrosine. And this is also validated by the alanine scanning. If we introduce the alanine to this aspartic acid position, and then the activity will be abolished. At the same time, the hydrophobic call over here is also coordinated with the tyrosine incorporation, or as a tyrosine as a substrate. So with this data, we're still curious, right? We're screening different enzymes. Is there any other enzyme? People show there's a lot of sulfotransferase. They can carry out a similar reaction. So that's why we did a structure similarity search, and we pull out all the enzyme has a similar structure with this sulfotransferase from IBase. And what you will see, the top hit is from mouse, from human, and they have really good score. But when we test their activity, you can see it's really stunning. It's like our sulfotransferase from IBase, they have really good activity, but from the other sulfotransferase, the activity is very, very low. And to explain this, we performed the docking study for all these structure. And the short answer is, if you look at our structure from this IBase salt T1C1, the tyrosine is very close to this PAPX, and it had the right position, this phenolic alcohol. It had the right position for this SN2 attachment. For the other structure, you can see the alcohol, the tyrosine is easy rotated. It's not at the right configuration, or maybe the distance, it will be a little bit far away. With this study, with these cells, with the sulfotyrosine or biosynthetic sulfotyrosine in hand, we are thinking about, is there any biomedical drug we can make using these cells? That's what we turn to the thrombin inhibitor, and people show different thrombin inhibitor with this sulfation. They can enhance their inhibition activity, and they can stop this kind of blood clotting. Richard Penn group has demonstrated a lot of good example for this, and they show the sulfur tyrosine is important for a lot of critical residue of this inhibitor. So we are using one of the model protein is the modani. And if you look at the co-crystal structure of this modani and the thrombin, the green structure is the modani, and the big structure is thrombin. And you can see it's very clearly this tyrosine 32 and the tyrosine 35. They go to this kind of negative charge binding pocket of the thrombin. And if people can incorporate the sulfate group over here, sulfotyrosine over here, and probably we will achieve some enhanced binding. And this is true from the IC50 data we did with the thrombin activity. And the gray curve is the wild type modani thrombin inhibitor. And if we introduce one sulfotyrosine at a 32 position, you see some enhanced activity. And if we introduce two sulfotyrosine to the modani, and we see a really potent inhibitor over here. So to summarize this part of the work, we created the cells with the site-specific post-translational modification, and we show that we can create these cells for both eukaryotic and prokaryotic, and they can serve as a potential drug factory to make these different drugs with enhanced therapeutic efficacy. And from 2017, my lab has been focused on working this idea. We created different cells for prokaryotic and eukaryotic. And some of the cell amino acid building block that are quite interesting. For example, this amino acid dopa, that the modification for the muscles. Why muscle can attach a lot of surface? Because they have this dopa moiety. And we can incorporate this dopa into different organisms. In that case, hopefully we can create some different cells. They can attach to the surface, and we can do a lot of different material or different biotechnology application using these cells. For the acknowledgement, most of the work I discussed over here was done by my graduate student Yuda and Guanlin Rui Mengxi. And this is our funding agency, and we are hiring. If you want to do some synthetic biology work, translational work, feel free to contact us. And thank you for your attention. I'm happy to take any questions. Thank you.
Oh, amazing talk on
Thank
very,
you.
very important topic. Actually, I love it. And also, I believe, I mean, many people will apply now because this video will be released, and then this is amazing, work. I mean, many people will kind of fascinated, of course, I mean, by this work. So thank you so much. So let me check any question from the audience. Okay, I will ask one question. So with your amazing data and advances in many protein structural kind of prediction tools like the AlphaFold and Rosetta and so on, I see great future in this field. But at the same time, there are many remaining challenges in this field. So could you kind of share your perspective on future opportunities and challenges in this amazing field?
Yeah, so I think, I'm not trained by the computational biologist or chemist, but when I started my independent career, I realized the powerfulness to combine both computational strategy with the experimental strategy. So that's why I start to bring them together. I don't think like one field they will solve the problem or like solve the goal or achieve our goal. I think it need to be combined. So that's why we start from bioinformatics, right? When we have no activity and they bring us some of the activity, and then we can use different experimental skill to bring that up. And also, I think this is kind of vice versa. For example, right now we start from the bioinformatics, and then we created a cell over here. And right now we have this cell, and you can think about right now we can come back over here to the enzyme evolution, right? Because we have the cell over here, we can couple this gene with the antibiotic marker, and we can combine to make this enzyme better, right? So I think it's a really kind of interdisciplinary, it's a help with each other. So that's why in my lab, I really hope to integrate different kind of technique. I don't believe like you only work with AlphaFold, you can solve the problem, you can get the most accurate structure, or you get the best enzyme. I don't think you can go over there. But once you combine these things and then you go forward, reverse several wrong, I think you will be able to find the best thing or the things you want. Yeah.
That's absolutely wonderful answer. So thank you so much. And then let me check one more time. So actually we do not see. So and then also we have only 1 minute left. So I close, and then we will have another session. So thank you so much one more time, and thank all for joining today and staying today. Although many amazing or very busy kind of schedule, especially during this last kind of time of the semester. And then again, thank you so much for all the audience and the speakers today. And we will meet again next week on May 5 to Thursday, again, the same time, the same Zoom link, but I will be joining all of you from New Orleans because I'm attending another conference, and we'll have the Professor Alfonso Yaramillo at University of Valencia, Spain, and Eric Young at Worcester Polytechnic Institute. And as usual, the follow-up informal chat will occur without recording. And please stay here if you are interested in chatting with today's speakers. I'll promote you to panelists who can speak and show your handsome and pretty face. And thank you so much. And I've stopped recording. Let me stop. Okay, stop.